وبركاته. المحاضرة الثانية من محاضرات مادة Microbiology section Bacteriology. Our lecture is about antimicrobial chemotherapy using of antibiotic for treatment of bacterial infection. Chemotherapy as a science began in the first decade of the 20th century by several attempts of researchers to treat bacterial infection. This is associated with understanding of the principles of 4 categories. There are 4 important categories in the subject of antibiotic or antimicrobial chemotherapy. The first is selective toxicity. Selective toxicity means the antibiotic should be affect bacterial cells or microbe cells, but not affect on host cell. This is very important point in the treatment of infections. Also, the second category is the specific chemical relationship between microbial pathogen and the drugs, where the drugs is act on cell wall or on cell membrane or on prevention of protein synthesis and others. Also, the most important category is the development of drug resistance. This worldwide problem now associated with a high rate of morbidity and mortality. Bacteria can be developed resistant to some antibiotic rapidly, which lead to a problem in the management of different types of infection. The fourth category is the role of combined therapy. Combined therapy means more than one antibiotic to treat the infection, as seen now, because of the development of drug resistance. You should be used several antibiotics, such as 2 or 3 for treatment of several infection, especially in the bacteria that known to be produced or develop resistance rapidly to antibiotics, such as E. coli and staph aureus. Now, we study the range of action of antibiotics. Antibiotics fall into 3 main categories. As you know, we can classify bacteria into 2 groups: gram-positive organism and gram-negative. This depending on the structure of their cell wall. So antibiotic may be action against gram-positive organism only, like penicillin, erythromycin, and vancomycin, or action mainly against the gram-negative organism, such as polymyxin and nalidixic acid. Or the antibiotic able to kill or inhibit the growth of both gram-positive and gram-negative organism. This is called broad spectrum antibiotics, like ampicillin and ciprofloxacin. Now we study the mechanism of action of chemotherapeutic agents or antibiotic. The first and most important mechanism is the inhibition of cell wall synthesis, such as beta-lactams drugs that include penicillins and cephalosporins. Penicillins such as penicillin G, amoxicillin, ampicillin, and cephalosporin, including ceftriaxone, cephalothene, and cefotaxib. This interferes with the cell wall synthesis and causes bacteriolysis. Maybe bind with sugar or protein through the and prevent the transglycosylation and transpeptidation process that help in formation of cell wall. Also, inhibition of cytoplasmic membrane function, for example, polymyxin, or inhibition of protein synthesis, such as tetracycline, erythromycin, and aminoglycosides, which including tobramycin and gentamicin. This drug effect on 30th subunit of bacterial ribosome and prevent translation of mRNA to peptide or amino acid sequence. The third mechanism, fourth mechanism is the inhibition of nucleic acid synthesis, such as in case for quinolone. Most important antibiotic in this group is fluoroquinolone, such as ciprofloxacin, norfloxacin, and levofloxacin. This antibiotic effect on gyrase enzyme, gyrase B enzyme, that prevent folding of nucleic acid during DNA replication. Also, rifampicin, novobiocin, and sulfonamide act on nucleic acid synthesis. The fifth mechanism is competitive inhibition, in which the drugs compete with essential metabolite for the same enzyme, as in case of sulfonamide. Sulfonamide resembles to para-aminobenzoic acid, and it's entered to the reaction instead of PA, and prevent DNA synthesis. Now, any question about the mechanism of action of chemotherapeutic agents? Any question? Ahmed Jabbar, Ahmed Jabbar, Ahmed, تسمعني؟ أحمد علي محسن.
نعم دكتور موجود.
تسمعني؟
أي نعم أسمعك.
أكو سؤال واضحة هذه
لا دكتور
الميكانيزم؟ واضحة إن شاء الله؟
إيه.
Now we study mechanism of resistance to antimicrobial agent. This is very important problem in the management of bacterial infection associated with mortality and morbidity. This act due to prolonged use of antibiotic and bacteria able to develop resistance rapidly. The first mechanism is the ability of organism to produce enzymes that degraded the drug, such as the production of beta-lactamase enzymes that destroy penicillin by penicillin-resistant staphylococci. Staphylococci, such as Staph aureus, able to produce a variety types of beta-lactamases enzyme and can be destroyed wide range of antibiotic by degrading of beta-lactam rings that presents in the structure of antibiotic and lead to development of resistance. Also, enzyme acetyltransferase produced by gram-negative bacteria, which can be destroyed chloramphenicol. The second mechanism for bacterial resistance is the organism change its permeability to the drugs by modification of specific protein in the outer cell membrane. This means the antibiotic unable to enter to the host, to the bacterial cells. This occurs due to modification or alteration in the surface protein of the bacterial cells that presents in the outer membrane. So there is no receptor for antibiotic and prevent entering of antibiotic to the cell, such as in case of resistance to polymyxin. Also, the organism develop an altered receptor site for the drugs. Each substances or each antibiotic, each toxin have a specific receptor in the cell membrane or cell wall of bacteria. But when this receptor is altered, maybe due to mutation, such as deletion or insertion, this leads to alteration in the protein or receptor configuration and prevent binding between antibiotic and this receptor, so this antibiotic is become inactive. For example, alteration of specific protein in the 30S subunit of bacterial ribosome that serve as a binding site for in the susceptible organism for many antibiotics such as aminoglycoside, including amikacin and gentamicin. The fourth mechanism, the organism develop an altered metabolic pathway that bypasses the reaction inhibited by the drugs. For example, sulfonamide resistant bacteria acquire ability to use performed folic acid with no need for extracellular para-aminobenzoic acid. This figure shows the bacterial structures that as a target for the antibiotic and the types of antibiotic that affect on this part, such as in cell wall synthesis, we see several types of antibiotic able to degrade this structure, such as cycloserine, vancomycin, bacitracin, penicillin, cephalosporin, carbapenems, such as meropenem and imipenem, and others. Now we should be what's the origin of antibiotic resistance? How this origin is developed, causes of this development. There are two important or general cause for drug resistance. The first is non-genetic, while second is genetic drug resistance. Non-genetic drug resistance include metabolic inactivity. This means the microorganism become inactive. So some antibiotic is not active against non-active bacteria. Most antimicrobial agents effectively act only on replicating cells. Non-multiplying cells are resistant to drugs, such as in case of Mycobacterium tuberculosis that associated with the tuberculosis, which survive for years in tissue, especially in the microphage, or in the granuloma inside the lung, and remain maybe for decades. But if this bacteria has become active, this become susceptible to antibiotic. This occurs after the suppression of cellular immunity of the patient. As you know, the main type of immunity that control mycobacterial infection is the cellular immune response. And when the cellular immune response is shifting to the humoral immune response, this leads to activation of mycobacterial tuberculosis and developed of signs and symptoms of disease. But at the same time, the bacteria become susceptible to antibiotic that used to treat it of infection. The second mechanism is the loss of target structure. Protoblast or all forms of bacteria are penicillin resistant because they lost their cell wall, which is a structural target site of the drugs. For example, as we saw in the first lecture, mycoplasma. Mycoplasma originally has no cell wall. So any antibiotic that affects on beta-lactam ring, such as penicillin, cephalosporins, carbapenem, are not effective against this bacteria. We are able to use erythromycin or tetracycline. The third cause of non-genetic drug resistance. Bacteria may infect the host and the sites where antimicrobial are excluded or not active or cannot be reached. For example, aminoglycosides such as gentamicin, amikacin, tauromycin are not effective in treatment of typhoid fever or enteric fever, which causes by Salmonella typhi. Because of this, bacteria are intracellular and aminoglycosides do not enter to the cells. Now we study the genetic drug resistance. This occurs due to three important mechanisms. The first is plasmid-mediated resistance. The plasmid is an extra-chromosomal genetic element. Can be divided into two main types. Fertility plasmid and resistant plasmid. Fertility associated with the production of some protein help in the growth of cells or ability to produce the virulence factors such as toxins, while R plasmid or resistant plasmid is carry antibiotic resistant gene to the host cells. The plasmid is self-replication. The replication of a plasmid is independent to the replication of bacterial chromosome. So plasmid carry many genes that code for production of enzyme that destroy or inactivate antibiotic. For example, beta-lactamases that destroy beta-lactam, which are produced by gram-negative bacteria, especially E. coli and Pseudomonas aeruginosa. A plasmid also may result in epidemic resistance among bacteria by moving from one to other by a process called conjugation. Conjugation between two bacteria help in the transmission of genetic material, especially plasmid, between one species or one bacteria to another. Other genetic drug resistant mechanism is transposon mediated resistance. Transposon is a segment of genetic material, or DNA, carry gene that code for drug resistance. As they move between plasmid and chromosome, they can transfer this property to bacteria. The transposon able to integrate it either in chromosome or integrated in the plasmid and added a new property for this bacteria. This process called transposition. The third origin is chromosomal drug resistance. This developed due to the spontaneous mutation in a gene that controls susceptibility to an antimicrobial agent. When a mutation is altered, the gene expression and maybe prevent the production of protein or lead to production of an active protein. شبابنا لحد الآن أكو سؤال؟
عفوا دكتور.
نعم.
دكتور عاد شوية توضيح أكثر.
توضيح علما اللي نقطة منهم؟
هم كل السلايدات يعني عاد أمشي بس بعد توضيح أكثر أريد.
هسه الجينيتيك دراج ريزيستنت؟
أي وأريد التوضيح يعني شوية أبسط يكون.
أبسط ماشي أوكي إن شاء الله. شباب احنا عندنا نوعين من انواع او اصلين اساسيات لتطور الانتيبايوتيك ريزيستنس. الاول اللي هو الفيرست اللي هو نون جينيتيك دراج ريزيستنس، يعني ما له علاقه بالقضايا الوراثيه، وهذا يشمل include 3 points. First, metabolic inactivity. Metabolic inactivity. بعض الانتيبايوتيك يكون active فقط ضد الريبليكيتنج سيلز، البكتيريا اللي تكون نشطه وناميه الانتيبايوتيك يكون فعال ضدها. اما النون مالتيبلاينج organism or an active bacteria this resistant to this drugs. وقلنا انه كما في حاله Mycobacterium tuberculosis. Mycobacterium tuberculosis may remain for years inside the lung tissue, and this bacteria is not susceptible to antibiotic used because of this bacteria presence in the dormant state، يعني تكون في حالة خاملة. السبب شنو هنا بهاي الحالة؟ انه infection is controlled by cell-mediated immune response. But when suppression is occurred to the cell-mediated immune response, this bacteria is become active and replicate. So this bacteria is become susceptible to drug. السبب او الاصل الاخر هو loss of target structure. كل انتيبايوتيك له هدف معين في الخليه، مثل ما عرفنا الان بالسلايد السابق انه some antibiotic act on cell wall of bacteria, some antibiotic act on ribosome, prevent protein synthesis. Some antibiotics such as fluoroquinolone active or prevent nucleic acid synthesis. فكل واحد من هاي الانتيبيوتيك له فد تارجت معين. إذا كان هذا التارجت مفقود، البكتيريا راح تكون مقاومة لهذا المضاد المعين. وانطينا مثال في المحاضرة السابقة كما في حالة بكتيريا المايكروبلازما. Mycoplasma, the unique feature of this bacteria, Mycoplasma has no cell wall. Mycoplasma ما عندها cell wall، وبالتالي راح تكون مقاومة لجميع الانتيبايوتيك أو المضادات الحياتية اللي تعمل على منع السيل وول synthesis or peptidoglycan synthesis. Also, the third origin, bacteria may infect the host in the sites where antimicrobial are excluded or not active. يعني راح تصير البكتيريا في موقع معين، هذا الموقع لما يدخل الانتيبايوتيك راح يطرد بسرعة من هذا المكان أو من موقع الإصابة، وبالتالي there is no contact between bacteria and antibiotics. This non-genetic drug resistant origin. While genetic drug resistance, this depending on the presence of genetic material, different types of genetic material. أهم ما موجود عندنا هو البلازميد mediated resistance، المقاومة المرتبطة بالبلازميد. Plasmid is a circular extra chromosomal genetic elements. هي عبارة عن genetic elements DNA يكون خارج الكروموسوم، ما له علاقة وين بالكروموسوم. Self-replicating, not dependent on chromosome replication. يعني يكون مستقل عن الكروموسوم. هذا البلازميد أو هذه genetic material able to carry antibiotic resistant gene، جينات المسؤولة عن المقاومة، فتنقلها إلى داخل الخلية ويصير expression for the gene or this gene and lead to development of antibiotic resistance. This usually occur in gram-negative bacteria and especially in case of beta-lactam drug, because of the plasmid carry the gene that is responsible for production of beta-lactamase enzyme. Genetic material carry the gene that is responsible for drug resistance, but this structure is integrated to the plasmids and or chromosomes and transfer this property to bacteria. وبالتالي راح تنتقل هذه الصفة أمنيا إلى البكتيريا، لأنه راح يدخل الترانسبوسوم في داخل المادة الوراثية لهذه البكتيريا. هذه العملية نسميها شنو؟ ترانسبوزيشن. والكروموسومال دراج ريزيستنت هي تحدث في الكروموسوم، في الجينات المسؤولة عن المقاومة الموجودة بكروموسوم الخلية أو DNA الخلية، والسبب هنا هو السبونتينوس ميوتيشن، حدوث طفرة في الجينات المسؤولة عن السيطرة على المقاومة للمضادات الحياتية. شباب صار واضح هذا الشيء؟ شباب اكو سؤال؟
لا هسه واضح دكتور عاشيله.
هسه راح نجي الى موضوع مهم هو complications of antibacterial chemotherapy، المضاعفات الناتجة عن الكيمو antibacterial chemotherapy. الأولى هي development of drug resistant، يعني احنا لما ننطي antibiotic راح نتوقع انه موجودة بعض الcomplications أول وأهم complications هو development of drug resistant، تطور المقاومة للمضادات، و side effects or the second complication is drug toxicity. Some drugs have a toxic effect for host cell، وعندنا super infections. Fourth is hypersensitivity. development of drug resistance هاي حكينا عنها قبل شوية. طبعا أيضا من الأسباب المهمة لتطور for development of drug resistance is the prolonged use of antibiotic. وهذا شايفينه حاليا بمجتمعنا انه الاستخدام طويل الامد للمضادات الحياتيه راح يؤدي الى ظهور عزلات مقاومه للمضادات المستخدمه. ايضا عندنا drug toxicity. Some antibiotic has a toxic effect against host cells, such as tetracycline. Tetracycline lead to decrease the bone development. يعني tetracycline لما نعطيه هسه يعتبر contraindication بالنسبه للاطفال ما ينطى، لانه يؤدي الى تاخر في نمو العظام والاسنان. عندنا ايضا السوبر انفكشن. السوبر انفكشن the infection with the antibiotic resistant bacteria. مثل ما معروف انه احنا لما نعطي الانتيبيوتيك، فالانتيبيوتيك هذا ما راح يكون فعال ضد بكتيريا دون اخرى، وبالتالي ايضا راح ياثر على النورمال بكتيريال فلورا الموجوده في موقع الاصابه. وبالتالي لما نستخدم when we use the antibiotic is lead to suppression or killed susceptible organism such as some normal flora, while the resistant bacteria is remain live or not inhibited and lead to development of super infections. يعني احنا عندنا اكو بكتيريا طبيعيه نورمال فلورا موجوده في جسم الانسان، كما موجوده in skin and the intestinal tract colon. فاحنا لما نستخدم الانتيبيوتيك، قسم من الانتيبيوتيك راح يؤدي الى قتل هذه البكتيريا، والبكتيريا المقاومه راح remain available, so lead to development of super infection. مثل ما معروف انه النورمال بكتيريا الفلورا الموجوده بالامعاء مثلا causes competition with pathogen، راح تتنافس مع الباثوجين على الغذاء او غيره، and also able to produce some toxin or antibacterial substances that lead to kill pathogen، لانه راح يكون اكو التنافس على الغذاء والمواد الاخرى، وبالتالي هذا راح يؤدي قتل النورمال فلورا، killing of normal flora or decrease the number of normal flora, lead to development of super infection due to drug resistant bacteria. The fourth complications is the hypersensitivity. This means development of anaphylactic shock or type 1 hypersensitivity, as we seen in the case of penicillins or sulfonamide. يعني المريض نشوفه يتحسس بسرعه مثلا في حاله البنسلين، استخدام البنسلين او استخدام السلفوناميد. نشوف تطور حاله الهايبر سنسيتيفيتي اللي هي التايب 1 هايبر سنسيتيفيتي maybe reach to the anaphylactic shock، ممكن انه تؤدي الى anaphylactic shock, coma and death. واضحه هذه شباب الكومبليكيشن؟
نعم دكتور.
اي دكتور.
واضحه؟ عندنا 4 مضاعفات اساسيه طبعا اللي هي development of drug resistant وحكينا عنها، والدراج توكسيسيتي يعني بعض الانتيبيوتيك تكون toxic for patients or have a toxic effect on the host cells. وعندنا السوبر انفكشنز، وهاي قلنا السبب هو انه لما نعطي انتيبيوتيك راح نقتل البكتيريا الطبيعيه الموجوده. طبعا البكتيريا الطبيعيه الموجوده راح تسوي كومبوتيشن ويا الباثوجين حول الغذاء، ولها قدره على انتاج بعض الانتي مايكروبيال ايجنت اللي تثبط الباثوجين او تقتله. فلما احنا نستخدم الانتيبيوتيك ونقتل النورمال فلورا، البكتيريا او الباثوجين اللي يكون resistant to the antibiotic is grow and lead to development of infection. والسبب الاخير هو الهايبر سنسيتيفيتي. مثل ما تعرفون انه بعض الاشخاص تكون عندهم حساسيه للبنسلين او للسلفانومايد، وبالتالي راح تتطور حاله الانفايلكتيك او تايب 1 هايبر سنسيتيفيتي، بحيث انه تظهر الحساسيه بسرعه خلال دقائق. شباب اكو سؤال عن المحاضره اليوم؟ اكو سؤال عن المحاضره السابقه؟ اكو سؤال شباب؟
لا دكتور ماكو، شكرا جزيلا.
مصطفى
نعم دكتور.
اكو سؤال؟
لا دكتور واضح.
المحاضره واضحه؟
نعم دكتور ان شاء الله.
احنا طبعا هي هاي الخطوط العريضه وهي هاي المطلوب من عندكم انتم. بس اريد من عندكم شويه تقرونا عن في الكتب المنهجيه الموجوده اللي قلنا لكم عنها، انه جاوس موجود عندنا. تقرونا عن الانتيبايوتيك، يعني تعرفون كل انتيبايوتيك كل جروب، يعني المهم الجروب بصوره عامه، لان هي الصفات مالت الجروب تنعكس على صفات الفرد. فاريدكم تشوفون تقرون الانتيبايوتيك وتعرفون كل انتيبايوتيك وين راح يشتغل، التارجت مالته، و الكومبليكيشنز. طبعا بالنسبه للسايد افكتس كونترا انديكيشن غيرها، هاي انتم راح تاخذوها بالفارماكولوجي، احنا مو ويانا الان. اكو سؤال شباب؟ اديان حيدر؟ ماشي. شكرا جزيلا لحسن اصغائكم، و thank you very much for